New insights into autism spectrum disorder. 7 genes that may increase risk. Autism spectrum disorder, ASD, is a complex neurodevelopmental condition characterized by challenges in social interaction, communication, and restricted or repetitive behaviors. The genetic underpinnings of ASD have long fascinated researchers, leading to the discovery of numerous genes associated with the disorder. Recently, studies have identified several new genes that may contribute to an increased risk of developing ASD. This video will explore 7 of these newly identified genes and their potential implications for understanding ASD. Number 1. Plextrin homology domain containing A8 is a gene that plays a role in cellular processes, including cell signaling and cytoskeletal organization. Recent research has shown that mutations or alterations in this gene are linked to a heightened risk of developing ASD. The gene's involvement in cell signaling pathways is critical, as these pathways are essential for neurodevelopment and synaptic function. Dysregulation of this gene may affect brain connectivity, which is often altered in individuals with ASD. Number 2. Vesicle transport protein is another gene implicated in ASD. It is part of the vacuolar protein sorting, VPS, which plays a crucial role in the transport of proteins within cells. Disruptions in this gene function may lead to improper trafficking of proteins essential for neuronal development and communication. Studies have shown that mutations in this gene can lead to impaired synaptic transmission and contribute to the development of ASD phenotypes. Number 3. Lysine methyltransferase 2C is a gene that encodes a protein involved in the regulation of gene expression through histone modification. Recent findings indicate that mutations in this gene are associated with an increased risk of ASD, suggesting that epigenetic factors may play a significant role in the disorder. The gene is thought to be crucial for proper brain development and function, and disruptions can lead to neurodevelopmental anomalies. Number 4. Homology and guanine nucleotide exchange factor 3 has also been linked to ASD. This gene is involved in synaptic transmission and neuronal communication. Alterations in this gene may affect the formation and stability of synapses, which are critical for efficient signaling between neurons. Both genetic and environmental factors could potentially contribute to changes in gene expression, leading to an increased risk of developing ASD. Number 5. 5-hydroxytryptamine receptor 2A is a gene that encodes a receptor for serotonin, a neurotransmitter implicated in mood regulation and social behavior. Variants in this gene are associated with several neuropsychiatric disorders, including ASD. The role of serotonin in neurodevelopment has been widely studied, and alterations in this pathway can significantly impact social behavior, communication, and anxiety levels in individuals with ASD. Number 6. Killer cell lectin-like receptor D1 is primarily involved in the immune system, particularly in the response of natural killer cells. There is growing evidence suggesting that immune dysregulation may play a role in the etiology of ASD. Variations in this gene may influence immune responses and inflammation, which in turn could affect neurodevelopment and contribute to the risk of ASD. Number 7. Solute carrier family 6-member 4, which encodes the serotonin transporter, has long been studied in relation to mood disorders. New findings indicate a potential link between this gene and ASD. Variants in this gene may impact serotonin reuptake, leading to altered levels of this crucial neurotransmitter during critical periods of brain development. As serotonin is essential for various neurodevelopmental processes, disruptions may increase susceptibility to ASD. The identification of these 7 genes represents significant progress in understanding the genetic landscape of ASD. The complex interplay between genetic and environmental factors suggests that each individual's risk profile is unique. This knowledge paves the way for more targeted research efforts and potential therapeutic interventions. Future studies will be essential to elucidate the precise mechanisms by which these genes influence neurodevelopment and behavioral outcomes. This will not only enhance our understanding of ASD, but also inform the development of personalized treatment strategies and interventions tailored to the genetic profiles of individuals with ASD. In summary, the discovery of new genes associated with an increased risk of autism spectrum disorder highlights the intricate genetic architecture underlying this complex condition. As research progresses, it is hoped that a clearer picture of the biological processes involved in ASD will emerge, leading to improved diagnosis, treatment, and support for those affected by the disorder.